Our talk. We are very excited to give our presentation for the DNA Sequencing Research Group. We have a couple updates to our studies. We're going to give a update to our last year's study, a single cell project, and then our current study, which is a DNA methylation project. But first, we're going to go ahead and get started with one of our sponsors. Thank you so much to Watchmaker Genomics. They're going to go ahead and give a brief talk. That's going to be followed up by our lightning talk by Brennan Grimes. And then following that, we'll have Charlie Whittaker give a talk and update on our single cell project. And then we'll have Molly Zeller give kind of an overview of our study for the DNA methylation. And then we'll have Xiaoyu Zhuo give a study update on the analysis portion. So first, I'd like to welcome Ariel Hannick to the stage, and yeah, please give her a hand, and thank you so much, Watchmaker, for your support.
Good morning, everyone. I'm Ariel Hannick, a product manager with Watchmaker Genomics. We're a genomics tools company headquartered in Boulder, Colorado, with over 130 employees worldwide. Our focus is on engineering enzymes and tailoring solutions designed to address gaps or needs across a variety of challenging applications, all with a focus on improving data quality. As I know you're all aware, core labs and sequencing service facilities face a unique set of needs, namely maintaining high performance across a variety of sample types, variable inputs and quality and sources, price, where there's a constant drive to reduce operational prices through a combination of reducing reagent costs and maximizing sequencing economy, as well as workflow, finding solutions that are easy to implement, easy to automate, have rapid turnaround time, and low hands-on times, are all hallmarks of solutions that will ultimately drive down laboratory costs. It's often the case that these areas are in direct opposition with one another, where you can sometimes find two in a solution, but typically not all three. Watchmaker is working to change that. We offer a complete NGS portfolio that includes DNA and RNA library preparation kits, as well as library amplification and automation solutions. And we're going to try and touch on all of these today. Our DNA library prep kits are available both with and without enzymatic fragmentation. Both kits support each kit features a rapid workflow, supports PCR-free sequencing, and enables inputs as low as 500 picograms. Starting with our DNA library prep kit with fragmentation, we look at this as enzymatic fragmentation being a scalable solution that can avoid the equipment and consumable costs typically associated with sonication. But we also recognize that there have traditionally been gaps in terms of ease of use and implementation of enzymatic frag. Our library prep kit with fragmentation overcomes these pain points. At left, we highlight tunability, demonstrating that library size can be easily modulated through a combination of fragmentation time and temperature, achieving consistent results between replicates. At right, we use the same fragmentation conditions across a 5,000-fold input range from 100 picograms to 500 nanograms, achieving nearly identical library sizes, and demonstrating that you can use the same fragmentation conditions for all sample inputs. Another traditional pain point of enzymatic fragmentation is the increased presence of sequencing artifacts. The overabundance of these artifacts convolutes identification of true structural and single nucleotide variants. What you can see with the Watchmaker chemistry shown in purple is that we have many fewer of these artifacts relative to competitor kits and have reduced the level to nearly on par with sonication, eliminating the excess artifacts. Of course, not all samples require fragmentation, and for samples that don't require fragmentation or have gone through a mechanical fragmentation process, we have our DNA library prep kit, which features a tailored ligation to maximize conversion of DNA to sequenceable library. In this example, we use 1 nanogram of real patient-derived circulating free DNA as input into 3 different library prep chemistries. The Watchmaker chemistry results in 9-fold higher library yields and resolution in both the monosome and disome peak. The improved conversion has a meaningful impact on sequence coverage. Here, we're looking at cumulative coverage data compared to other chemistries, all sequenced to equivalent read depths. The shaded region shows the increase in percentage of bases covered with Watchmaker at a given depth X. This translates directly to sequencing economy, as fewer reads are required to achieve a given target coverage depth. On the RNA side, we seek to have solutions that have the same similar features, robust performance, including with challenging and limiting samples, as well as streamlined and automatable workflows. Our core RNA library prep kit can be coupled with either mRNA capture or Polaris depletion, depending on project and sample needs. Either workflow is under 5 hours, enabling a truly single-day RNA library prep. We start with a snapshot of our RNA library prep workflow with Polaris for ribosomal RNA and globin depletion. Note that reaction steps are combined and shortened, and the number of spry bead cleanups is reduced. In total, this saves approximately 2 to 3 hours and 1,000 pipette tips per 96 libraries. Additionally, significant attention is dedicated to processing FFPE samples, including implementation of a novel treatment set that helps reverse residual crosslinks in FFPE material, enabling more RNA to be available for cDNA synthesis. We first look at performance with blood-derived RNA inputs ranging from 500 nanograms to 1 nanogram. We can see that as input drops, so does sequencing economy, typically due to an increase in pass filter reads that are unaligned, as well as reads mapping to ribosomal RNA in globin regions. This is true for all the chemistries evaluated, but the degree to which it occurs is much less with the watchmaker chemistry, especially at the lower inputs. The reduction in uninformative reads translates to improvements in unique gene detection sensitivity, shown at right, where you can see that watchmaker detects more unique genes, especially with inputs less than 100 nanograms. With respect to FFPE and low input performance, we look at gene detection in a matched fresh frozen and FFPE sample set. On the left, we look at the number of unique genes detected in 10 nanograms of each sample. The watchmaker chemistry is faster than competitor kits and results in greater unique genes detected. Moreover, the degree of overlap between the genes detected in the fresh frozen and FFPE datasets is greatest with watchmaker's chemistry, as shown at right, and this indicates that more biological information is accessed in FFPE samples when using watchmaker chemistry. Similar to our depletion workflow, our mRNA library prep kits are designed for rapid execution and simple automation. Notably, we employ on-magnet washes during mRNA capture, reducing handling and magneting time. In terms of performance, we first look at residual ribosomal reads, shown on the left. These should be low when using mRNA capture. Watchmaker has the lowest residual ribosomal RNA content and most consistent performance across the entire input range, resulting in more usable sequencing data. Additionally, more unique genes are detected with watchmaker, especially at inputs of 10 nanogram or less, again, indicating access to more biological information when sample input is limited. As we've seen repeatedly, when sample quantity is limited, library complexity often suffers in less unique data as well as data distortion. Here we compare libraries prepared from different inputs of the same sample using a differential expression analysis. With watchmaker, distortion is minimized and fewer genes are marked as differentially expressed, indicating higher confidence in the results when limited sample input is available. It's worth noting that all our library prep solutions leverage our high-efficiency, low-bias Equinox polymerase for PCR and are designed with automation in mind. Equinox was tailored specifically for fidelity to support highly sensitive applications. Here, we look at base misincorporation rates of Equinox polymerase relative to kappa hi-fi. Overall, Equinox demonstrates lower base misincorporation and significantly driven by the decrease in C to T substitutions, which are relevant for oncology applications. At this point, I've hopefully touched on the simplicity and sensitivity of our solutions, but we wouldn't be able to call this talk sassy without touching on automation. Each product in our NGS portfolio is built with automation in mind, including minimal transfer volume suitable for standard liquid handlers and generous overages to support dead volume requirements. Additionally, we have a dedicated automation support team who provides consultative support to users wishing to automate and optimize our workflows, They also work with a variety of third-party vendors across multiple automation platforms to develop off-the-shelf scripts that are available for download. And you can see we have a growing library of available methods, and this chart is constantly being updated. And with that, I'd like to thank you for your time and invite anyone with questions to stop by booth 509 or to join us tomorrow morning at 8 a.m. for Sequencing with SAS, the extended cut. There will be refreshments, including mimosas served. You're welcome to stop by.
Thank you, Ariel. And I want to say, we actually automated the Watchmaker RNA prep, so it's on our Hamilton, so it's a really great prep. And we're going to go ahead and move into our lightning speaker, and then we'll go straight into the DSRG talks. And so we'll hold all the questions until after we've gone through all the talks, okay?
Alrighty. Hello, everybody. My name is Brennan Grimes. I'm a member of the Genomics Core at the Van Andel Research Institute in Grand Rapids, Michigan, and I've been evaluating 2 recently introduced total RNA-seq workflows at low input. So until recently, there has been a lack of kits that can accommodate both low and standard inputs in the same prep. And our typical choice for a low input prep is the Smarter Stranded Total RNA-seq kit, version 3 for pico input mammalian samples. From Takara Bio, and that kit can accommodate 250 picograms up to 10 nanograms of input. Recently, however, 2 new kits have been introduced somewhat recently. And the first is the Watchmaker RNA Library Prep Kit with Polaris depletion from Watchmaker Genomics, and the second is the SparQ RNA-seq human, mouse, and rat kit from QuantaBio. And you'll see both of these kits can accommodate 1 nanogram up to 1,000 nanograms in input. So this flexibility was really appealing, and we wanted to see how these kits would perform at low input. To do this, we took 3 human total RNA samples of varying RIN quality and prepped 2 replicates of each at a range of inputs, and those inputs were 250 picograms, 1 nanogram, and 10 nanograms. We sequenced these to an average of 13 million reads, and our analysis compared the percentage of molecules that were uniquely mapped using STAR, percent of reads aligned using salmon, the library complexity, the percentage of nuclear rRNA reads, and the percentage of duplicate reads that we were seeing. I'm going to go through these graphs very quickly because this is a lightning talk. So looking at the proportion of uniquely mapped reads, up here in purple, the overall best alignment was seen with the Watchmaker Genomics kit, but this was not statistically meaningful at the higher inputs of 1 nanogram and 10 nanograms of input. Moving on to library complexity, in the middle in blue is the Takara Bio kit, and that kit had the greatest diversity. The same trend was seen with the percentage of duplicate reads, as you might expect. However, this was not statistically meaningful at the higher inputs. It was at the lower inputs, and this was regardless of the input quality. Finally, the proportion of nuclear rRNA. In pink, you see QuantaBio, purple is Watchmaker, and then in blue along the bottom is the Takara kit. QuantaBio and Watchmaker overall were less consistent throughout the different input amounts, and Takara Bio had the best ribosomal reduction, and this was statistically meaningful at higher inputs regardless of the input quality, and it was just overall more consistent across the different input groups. We did look at comparing between the different quality groups, but we found that overall this had much less of an impact, and the same trends were observed. Watchmaker had the best alignment, Takara had the most diversity and the least amount of duplicates, and additionally the least amount of nuclear rRNA. Overall, we came to the conclusion that regardless of the input amount or quality, libraries produced with the Takara Bio kit consistently had the highest diversity, the least duplicate reads, and the lowest proportion of residual rRNA. The Watchmaker Genomics kits and QuantaBio kits would require further optimization to be suitable for low input applications. And again, I want to point out that Watchmaker and QuantaBio were for 1 nanogram to 1,000, not 250 picograms, which we were using. And those protocols for both of those kits have since been updated since I've done this testing. I would like to give my acknowledgments to all the people at Van Andel that have helped us out, and of course to Watchmaker Genomics and QuantaBio for all your help throughout this process and enabling this project. Thank you.
There we go. Okay. Hi. So I'm presenting an update on the DSRG GBIRG collaboration. I presented on this in Boston last year, first phase of it, and I'll update some of the stories that we have to tell. Our experimental design is the first thing to think about. It breaks down into two, let's see, does my pointer work? It breaks down into two separate arms. There's the PBMC arm and a leukocyte arm. The leukocyte arm has 20 samples in it in two groups. One is the 10X platform. The other is the honeycomb platform. There are different time points. So in the 10X group, there was a 3' DGE component that was run at day 1, and then a set of 4-week fixed samples run on the Flex platform. In the honeycomb, the same prep was used in all the cases, but we also have 4 groups at 28 days and 1 group at 1 day. Each one of these samples was replicated, so a separate technician processed each sample at each location. So there are 2 replicates for each one. 4 different sites for 10X, 4 different sites for honeycomb. For the PBMCs, it was all just 1 site, 1 replicate, day 1 time point, but it was run on 4 different platforms: the 10X 3' DGE, the 10X Flex, Honeycomb Hive, and Parse Evercode. So the leukocyte prep, these are kind of special samples in that they have a large amount of low information content granulocytes in them. So they required a bit of a different processing than normal. This same sort of processing was applied to the PBMC honeycomb samples, but we just used standard vendor filtering for the other 3 samples in the PBMC arm. This kind of special processing involved very light filtering just to exclude basically empty drops, followed by integration, clustering, cell type assignment, and then this sort of elaborate process of debris exclusion to identify real cells as opposed to debris that was present in the mix. I spent much of my time in the Boston talk talking about this debris exclusion process, but what we ended up with was kind of a confident collection of real cells in each one of the samples. And the data that I presented in Boston was focused on analysis of that. But one thing that we really wanted to do and spent a bunch of time working on, Owen Wilkins in GBIRG in particular, was to downsample the data to about 25,000 reads per cell in order to have a nice uniform sequencing depth per sample, per replicate, things like that. So that's what we ended up doing, and that was the input into the final analysis that I'll show today. These bar charts just kind of indicate the sequencing process that I talked about. So we had a varying amount of reads generated in each one of these samples, varying numbers of cells in each one, and as a result, here in this the middle panel, or this panel here, the sequencing depth varied quite a bit per sample. Oops, excuse me. And Owen went in there and flattened that all out to 25,000 per cell. Another aspect of this work that I want to emphasize and point out is the work of Alex Lemans at Rutgers. The GBIRG people that have been working on this are all distributed around the country, working in different computational environments. And one of the challenges that we had was how do we all actually use the same software to do the analysis so our things can be comparable? And Alex solved this problem for us using containerized computing environments, Singularity Image in particular, that we could all download and run on our local computational resources and get around any kind of trouble. So we all got to use exactly the same software in the same kind of a way, which was very handy. And it's back when we started working on this project, this was a real problem for me, and I've now started using these Singularity images in my core's work on a regular basis in various different ways. So once we had the data downsampled appropriately, we repeated that analysis where we integrated the data, clustered, went through an extensive cell type assignment procedure. We started off with fairly high resolution clustering and went in there, annotated pretty high level of detail the different cell types, and Owen worked really hard on this as well. And then that cell type assignment was kind of flattened out into these more general categories that are shown on the right side. In the leukocytes, as I mentioned, there's a large population of these low information content granulocytes that are present there. That same sort of a process was applied to the PBMCs. Went through and studied each cluster and identified each cluster and then kind of flattened it down into more general categories. And you can see these gold granulocytes are much less abundant in the PBMCs. This analysis here just gives a very high-level overview, UMAP plots of all 24 replicates that are in play to make the point that each one of the samples, each one of the preps that we have in our thing integrated very nicely, and there were cells present in all the different clusters, all the different UMAPs. So the performance of all the kits as far as finding the different types of cells that are present in these samples was really high across the board. Based on this observation, and we saw this with the full data set prior to downsampling as well, we decided to present QC statistics by merging the A and B replicates from each one of the leukocyte experiments, and that's what's shown here. These are the number of genes and the number of counts and the mitochondrial ratio for the different cell types. And the story here, I think, is that the kits are pretty uniform. These QC statistics are similar with the different cell types across the board. There's some subtle differences in there that I'm going to return to. But QC performance at this level was consistent and reproducible. Next is a comparison to the FACS data. So Fred in DSRG collected FACS data for each one of these samples, and we wanted to compare cell type proportions. So these are the different cells that are present in each mix, and I think the story here is in general, the data that we're getting from the single cell platforms matches the FACS data quite closely. There are some kind of subtle differences that might be interesting. For example, a bit fewer granulocytes in the Hive and the Flex kits, which you may expect. But in general, the results are pretty similar to the FACS data. And another potential observation is maybe some more annotated monocytes in the Hive prep. The same sort of story for the PBMCs exists, and the QC metrics are consistent and uniform more or less across the different cell types and the different preps. And when you go and look at the PBMC data, cell type proportions compared to the FACS, again, the granulocytes are much less abundant, and things look pretty similar to the FACS data, with the exception of maybe the more monocytes called annotated in the Hive set, and EverCode, the parse EverCode is in that same camp, maybe at the expense of T cells here in this, but in general, quite similar to the to the FACS data. But we did have those experimental variables in the leukocyte that we wanted to dig into, the time of storage and the different platforms. And this correlation plot kind of gets at those things. You can see down here in the lower corner, the 3' DGE from 10X is grouped together, A and B replicate. Then you have the Flex 28-day samples grouping into a clade. The Hive 28-day samples group in a clade, and then in the upper corner, the Hive 1-day samples kind of separated from the rest. But there's some sort of interesting subtleties in the plot to think about. First, in the Hive replicates, these are all kind of uniformly associated, and the A and B replicates from the different sites don't cluster together. So the data from the Hive, from the different locations and the different replicates are just all kind of Hive data. But there is structure in the Flex data. There's sort of two clades at the 28-day Flex, site 1 and site 4 grouped together, and site 2 and site 3 grouped together. So there is something going on in the data in that way. And we wanted to dig into the sites and the A and B replicates in more detail, and we took a differential expression approach to do this. And Jyoti and Devender at Purdue did a lot of this work. I'm going to show two panels of these differential expression data. The top part of the panel is the violin plot of the QC data showing counts, total counts, but similar thing for the number of genes as well. And then the bottom part of it is the box plots of the log fold change data. If there's a horizontal line right near 0, it's indicative of very few genes with anything other than a 0 fold change. And if there's any kind of spread in the box plot, it means there's some differential expression there. I want to emphasize that the log fold changes that I'm showing here are all very close to 0. Just we're digging down into the weeds a little bit on the data. Nothing you would consider a really substantial fold change, or very, very, very few things. So the box plot down below, I'm not good at this pointing thing. The box plot down below is like, the first box plot is the A versus B comparison for the green violin plot, and so on and so forth. And out of this, there's only one A-B comparison that has any fold changes really, other than 0 fold changes, and that's the A versus B in the site 4 flex data, where a small number of genes are changed there. But in general, A versus B, almost no differences. The other is the site. This is a bit more interesting. First, I'll take the hive, which are over on the right-hand side of the plot. These different sites all have kind of flat box plots around 0, so very little differential expression in any of the pairwise comparisons at the site level for the hive 28-day data. The flex, on the other hand, this is that structure that I showed in the correlation plot. Comparisons that involve, say, group 2 versus group 1, or group 2 versus group 4, or site 4. We'll have fold changes. If you make comparisons between 1 and 4, or comparisons between 2 and 3, you have very little fold changes. So there's some kind of thing going on with the flex samples processed at different sites. And we dug into that some more, and John Priel did this work. The thing that we dug into had to do with library complexity that we found. And what we noted was in the flex 1 and 4 sites, there was substantially different library complexity. The library was more complex. Site 2 and 3 had a lower complexity library. John checked knee plots to look and see about cell loadings, and the fact that the knee appears at a similar barcode rank indicates that the same number of cells were loaded. And then in the lower corner there is a data showing comparison between the flex at site 1 and 4 with the 3' DGE data that was prepared at site 2. It's a similar result, and then those lower complexity samples as well. This led us to talk to DSRG, and DSRG folks went back and looked at metadata, experimental metadata associated with the different processings. And what we noted was these low samples did not require any kind of cell concentration. Any sort of handling or aggregation of the cells was done in swinging bucket rotors, so perhaps a bit of a gentler treatment of the cells. And the output of this processing was a little bit higher percentage of neutrophils in the data, which are low information content cells, low UMI per cell data. So there may be some experimental subtleties that can lead to different populations of cells coming out of the processing in these flex samples that appears in the analysis of the data as different populations of UMI per cell result. And that's what we're working on at the moment. We also had the opportunity to do a comparison between A and time, 28-day fixation versus 1-day fixation. The 10x flex and 10x 3' DGE are completely different kits. I'll touch on in a moment. So there's a lot of differential expression genes between 28-day and 1-day there. But in the hive, it was exactly the same prep, exactly the same type of a kit. There were very few substantially different genes in 28-day versus 1-day in the case of hive. So time of storage didn't seem to have a big impact there. To bundle the PBMC part of the experiment back into the data, this correlation plot here shows various different comparisons, and there's a strong site component to it. The EverCode is down in the lower corner with 3' DGE from both parse and from both PBMC and leukocytes, and then you have the clades of the flex day 28 and the flex from the PBMCs, the hive clade at 28 days, and then out at the end, which I think is kind of interesting, is the hive and data from day 1, PBMCs and leukocytes grouped together up in the corner. These PBMC data give us a chance to take a look at the different molecular biology that I just want to emphasize here. And this is just an example gene that's involved in leukocyte differentiation. But the point is that these 4 different kits that we took a look at are very, very different in their molecular biology of the 3' bias for the 10x 3' DGE. The flex data all aligns on the location of the oligos, whereas the last 2 are spread across the gene body more. And John also made a aggregation, a metagene type of a plot to look at this gene body distribution as opposed to the 3' DGE up in this upper part. So I would just like to thank the GBIRG single-cell RNA-seq team. I've mentioned a couple people in there, but we've also got Claire and Sarah from Barrow, and Sean and Maddie from the University of Delaware, and also the DSRG for all of their collaborations. It's been a great interaction. We've got some work to finish up on this project. GBIRG is always welcoming new members, so if any of this stuff interests you, we're available. Please get in touch with me. We're also working a little bit with NF-Core and NextFlow about deploying these workflows in core facilities, so if that's of interest to you, that's kind of the next thing we're going to move on to as we get finished with this single-cell RNA-seq project. So.
It's not on here. I don't know. Oh, just kidding. Now it's good. Yes. Okay. Escape works if you come across that issue. All right. So hi. Thank you, Charlie, for a great talk. I am Molly Zeller, and I am a member of the DSRG. I've been a member of the DSRG for I think like 7 years. I work at the University of Wisconsin-Madison, and I will be presenting the intro to our 2023 DSRG methylation study. So this is going to be pretty good. And I'm going to have a quiz question, kind of a trivia question for you coming up. So, and also I'm just super jazzed to be here because ABRF is awesome. All right. So the title of our talk is It Me You're Looking For? The Multi-Platform and Cross-Site Evaluation of Technologies and Methods Used for DNA Methylation Profile. I thought about putting a little snippet of this music video in. I don't know if any of you have seen it, but it's creepy. So I was not about to torture any of you. You guys can watch it on your own time. So first I want to start with thank yous because Xiaoyu's going to kind of come in a little bit later and talk about data. So I just want to get the thank yous out of the way and done with because I think, honestly, the DSRG, this is a super collaborative effort. It involves so many people, so many vendors. Like, it's kind of insane that we can make all of this work because the amount of people that are involved, it's crazy. So Jessica and Fred are chair and co-chair currently this year, and then these are the list of the people who are currently in the DSRG. We are also always probably accepting new members, and we're the coolest research group. Sorry, Charlie. But yeah, feel free to reach out to any of us. We're always welcoming. And I do want to give a quick little side note. So we had the new members meeting yesterday, and I kind of was there to represent the research groups, and I know a lot of people I was chatting with. I do want to shout out to research groups. They truly, honestly, are probably some of my best friends on this research group because we talk every other week. So if you are looking to meet new people in the field, you want to collaborate with others, please join. Okay, so I do want to start with kind of also thanking all of our vendor support. Without the vendor support, we honestly would not be able to afford what we do. We do a lot of sequencing, which, as you know, can be expensive, and library prep, and all of this. And what we need is we need that support from these vendors, and they're very supportive of all of us. And with this in mind, my little question for you today is, these next couple slides, keep in mind, how much do you think we have in vendor support this year? So kind of keep in mind our study design. I'm gonna come back, I'm gonna ask you all a question, and I hope somebody has got a good answer for me. And no, it cannot be from somebody from the DSRG. All right, so not only for the people in the DSRG is there a lot of us, but there's also a lot of us in our labs, right? So I'm at the University of Wisconsin, and I, got some people to join from my lab to do a lot of the work in our study, because we all collaborate, we all do this. So these are all of the kind of the helpers, if you must. So the people in black are kind of the lab, the wet lab people who did a lot of things, and then the people in green are all the bioinformaticians. So funny enough, we're a DNA sequencing research group, but we don't actually have any bioinformaticians on our group. Also, hint, hint. So if you want to join, we need somebody. So we right now are asking for a lot of collaborators, right? So we reach out and we basically are like, hey, can you spend some time analyzing this tremendous amount of data that we're just going to dump on you? Thank you, Xiaoyu. So just wanted to thank out. Also, I'm biased. I do want to thank Emily. She is here. She's going to hate me right now, but she is awesome. And she shipped all the DNA to 10 different sites, not only once, but twice, because we actually ended up making a mistake with our first dilution. So she shipped DNA to basically 20 different sites. And she's amazing. So please say hello to her if you find her. I can introduce you to. All right, so just a really brief, kind of very, very quick intro to DNA methylation. I'm not here to teach you about DNA methylation. My assumption is that you have some knowledge of it. So the kind of just a quick, quick little intro. So genetics, I like to say, is the study of the heritable changes in gene activity or function due to the direct alteration of the DNA sequence. And so we're thinking that of, point mutations, deletions, insertions, translocations. And then kind of in contrast, we think of epigenetics as the study of heritable changes in the gene activity or function that is not associated with any change of the DNA sequence itself. So it's more kind of the, we're adding on, or that methyl group gets added to the C and so on. So why are we interested? So the DSRG is here to do kind of studies, not to say who's the best, like what's the best vendor. It's not here to say, A is better than B or B is better than C. We're here to do this research for you so that you as a core lab don't have to do it yourself. You can refer to us. We can look at the really cool and exciting things, check them out, see what's easier. Because I know as a core facility, we work with, different organisms, different sequencing technologies. Not every core is the same, right? If they are, that would be really boring. So we're here to just kind of test. So with that in mind, our study this year kind of was a lot. This was, we bit off a lot, but thankfully to the vendors and all the people who are doing this, we created this really great plan. So what we did is we have I would say also two arms to it, So we took 4 DNA samples, and I'll get to that in a second. But what we did is we wanted to do whole genome sequencing looking at the methylation profiles. We wanted to do short read, Illumina, long read with PacBio and ONT. And what we did is for Illumina, we did bisulfite conversion, and we also did enzymatic conversion. So we did Zymo and Illumina with their library prep, and then we did Zymo and IDT, and then we also did NAB's MSeq. So we compared that, sequenced them all on 2 Illumina 10Bs. And then we also, remember, we're keeping these in mind, right, for my question a little bit later. Then we also did long reads. We did PacBio and ONT, which does not need conversion, right? We don't need to convert because we're sequencing that native DNA. We looked at that, and we looked at these 4 samples in duplicate with PacBio on the Revio and Oxford Nanopore on LSK114, or using LSK114. And we did this all, the sequencing was done at WashU, so thank you, WashU, for being very generous in your time. Thanks, Mike. So what we did is we wanted to look at the 4 different samples. So first we kind of, and I know this is last on the list, but our controls, right? So we have a fully methylated control from Zymo and a fully unmethylated control from Zymo. Those are kind of our 2 controls we wanted to look at, and we wanted to throw in 2 different human samples. So one, I kind of call the normal sample, right? It's 12878, which I'm assuming we're all familiar with, but I don't want to make that assumption. But 12878, it's just your human cell line. And then we also have MCF7, which is our breast cancer human sample. So we wanted to throw in something, that wasn't normal. I know I'm using that word, it's probably not kosher, so sorry. So kind of planning all of this was kind of a treat. So we had all this DNA, right? But now we had to separate it, not only send it to the 10 sites, we had to make sure what sites were getting what DNA in what form, okay? So we wanted to make sure that all the DNA was prepped all together so that we didn't have, somebody extracting DNA here, somebody extracting DNA here, or sourcing DNA from different places. So what we did at UW, we sourced all the DNA, we combined it, QC'd it, and then some of it needed shearing, some of it needed acoustic shearing, some of it needed mechanical shearing, and then once we had all of that, we aliquoted it out, sent it to the customer, sent it to the clients, the collaborators, I mean, and then the conversion was all done at the site itself, because what we wanted is we had 3 different sites per prep. We wanted to make sure that those sites treated that DNA kind of throughout the full prep. And then once again, once everything was prepped, everything was packaged up, sent to WashU for sequencing. Just a really quick excuse me, overview of the DNA QC. I just kind of want to show you briefly that we did start with very high molecular weight DNA. You can see our femto traces here, and we did have to shear for long reads just to kind of break it up and yes, we can sequence long DNA, but sometimes a super long DNA does not sequence the best. So we need to make sure that everything is within a really good sequencing size. And then I did put this little femto pulse down here of our short read shearing. You can't see the number, but it's sheared to about 400 base pairs. So we also did take a quick survey of kind of our short read library preps, just to kind of get a feeling of kind of, what was the overall ease of use for each kit. And this kind of just showcases the difficulty of the protocol. So the first graph there is the overall ease, and it looks like we have so scale of 1, 1 is easy, 5 is extremely hard. So I would say overall things look fairly simple. We are all experienced technicians who do a lot of these on a regular basis. Some of our sites actually had never worked with the prep before, so that can be, a challenge in and of itself. And then the next survey we also asked is the time spent on library prep. And so we have total hands-on time and then total time from start to finish. So it's very interesting that Zymo and Illumina has somebody must be very speedy. So I don't know which site is which, so this was very kind of blinded just to get an idea, an overall consensus of the time it takes. And then also total time from start to finish, which I think is relatively close. So with that, I'm gonna change it to Xiaoyu very shortly, but keeping all of this in mind, does anybody have an idea what the total value of vendor support we had this year was? Somebody said it. Hi, Claudia, 100K. Does anybody agree, disagree, think higher or lower? 40, oh, okay, well, Claudia, you were pretty much spot on. It was 102,000. So that is when you think about vendor support, like we truly need these people. I don't know like if our RG could come up with $100,000, right? So we truly need to work with these vendors, and they are so gracious, and they're actually very easy to work with, and we truly appreciate it. So with that, I'm gonna turn the mic over to Xiaoyu, and he's gonna tell you about all of the cool data that we've so far seen.
Well, first of all, thank you very much for Charlie give great talk and for Molly to set up the stage. And now I can't wait to share with you all the exciting results we are getting at. So still, before we go to the result, I still want to say why we are doing this. So there have been comparisons done before, look at different methods to evaluate methylation. But with our study, we have the most updated technology with most updated chemical. And we also have very interesting samples. We have 4 different samples, methylated, unmethylated, normal cell line or cancer cell line. And our method is going to be published, make it available to everybody. And also, we're going to provide some guidelines to how to do this properly. So now this is plan of bioinformatics, how we do analysis. So we are looking at 5MC in CBG context. So for some method, I mean ONT, we can look at hydroxy methylation, but we just clap everything together so we can have apple-to-apple comparisons. And then once we get all the reads, we align to HG38 and then do some basic QC and look at the coverage. And for short reads, we're going to evaluate a conversion rate because this is super important. And then we're going to compare how reproducible are different kits to each other, how reproducible are we between different sites, and how reproducible across different platforms. And yeah, let's get into it. So I have some quick slides about the method to evaluate methylation using long-read method. I assume everybody know what is the whole genome bisulfite or enzyme method. But for the long-read, for PacBio, it's using the polymerase kinetics. When I say kinetics, I mean you can imagine a polymerase goes through the DNA line and make synthesis and determine what is the base. But if it's methylated, the timing is going to be a little bit off, but it's very subtle difference. So I try to look at, with my eye, doesn't work. I have to go through some machine learning model. So, but apparently it works. So this is PacBio's method. But for nanopore, it's actually more straightforward, right? So it's you go through the pool, you measure the current change. And if it's methylated or if it's unmethylated, the current change is going to be some different. Still, it has to go through machine learning, so not something human eyes can do. And then how we process that, once we get the FASTQ from our production team, so we're going to trim them 50 base pair from both pair and reads, so very consistent. And then we're just going to use standard Bismarck aligner and also do the methylation calling. And I also want to say for the Zymo sample here, it's a non-directional, so we have to do all the alignment using non-directional too. And for HiFi and ONT, it's actually much easier. I won't say much easier because it's just the machine for review machine. It will come up with a methylation tag in the unaligned band file. And for ONT, it's also, they have a model. You just run through the model and get methylation core integrated in the band file. Now, let's go through some short read QC first. So it says whole genome bisulfite, but also includes the EMC, the enzyme method. But the process is very similar, so they are grouped together. So if you look at the library size, all of them are very consistent. The NEB actually a little bit higher. But if you look at the total number of reads, Zymo has the highest. So data is this. And then if we look at the quality, it's also pretty good. If you look at the Q30, it's also pretty good. If you look at duplication reads, duplicated percentage is also very standard. Nothing weird happening given the coverage we have. And then we map to the genome and we look at mapping metrics. And if you remember, Zymo has the highest number of reads, but the mapping efficiency is the lowest among three. So in the end, actually, we got very comparable coverage of all three short read method. And how about conversion rate? This is also very important. I mean, for conversion rate, both NEB and Zymo have higher than IDT in a little bit, and the highest is NEB. But you can see the difference actually not that big. So then how about HiFi, the PacBio's QC? So there's not really much QC we can do about this. You get the sequence and also the methylation is there. But for each library, we can consistently getting about 10 million reads. And if you map to the genome, it's 100% mapped. I don't know why it's 100%. And all of them are pretty long. So the average length is about 10 KB long. If you look at violin plot is also around 10 kb. The distribution is also pretty tight. And the coverage, because we have so many reads, for each library we can get close to 30x coverage. And for ONT, it's measured differently, but basically it's very comparable. We get very high yield for all the libraries. And if you look at N50, we can get from about 15 kb to 20 kb for all of them. And when we map ONT reads to the human reference genome, we can also get about 25 coverage on average. And the mapping metric is also pretty good, very close to 100%. And for the EPIC arrays, QC also looks pretty good. And then we're going to look at how we integrate them together to analyze. So first of all, I'm from WashU, and the lab I'm from, we developed a different browser. It's called WashU Epigenome browser. So if you're familiar with UCSC browser, it's very similar, but there's some difference. We like it better, especially for methylation-related research. So from here, you can see we have genome annotation here, and repeat mask code repeat annotation next. And then we have something we call methyl-Seq track. So in methyl-Seq track, we use this to represent methylation information at each CpG site. So it's a histogram, right? The histogram is going to represent the percentage of C is methylated. As you can see here, it's the blue bar there. So if you can see gray, that means background, that means unmethylated. But if it's blue, it means highly methylated. And then if you can see, there's also a moving black line. And that black line represents the read depths we get for that CpG site, for that position. And I don't have a mouse here, but if you move your mouse, hover there, it's going to show the pop-up box showing the combined depths and the CpG sites. So you can move your mouse around to see exactly what depths and the CpG percentage for each CpG sites. So after we set stage about our data hub, what does the data look like? So we have so many data, so I'm going to just use Zymos data as a representation. So as you can see, we have 4 different samples, right? From top to bottom, we have methylated MCF7, 12, A78, and non-methylated. And you can see they are quite different from each other. However, within each sample, between sites and across sites, we get very, very similar profile of methylation of this stretch, right? Of course, this is only Zymo data. What about others? I said I won't show all of them together, but I merged each technology, each platform together, and then show them together. So from here, we also have the 4 different tracks here. 4 different samples here. And for each sample, all 5 platforms are listed here. Still, you can see it's very reproducible across all the platforms. And I have the QR code here. If you can use your phone, use a QR code to go to the data hub, you can see the real thing on the browser, and you can move around. And I see everyone's trying to do that. But the point is, this is hosted in the lab's STP server. So if everybody is trying to access it, I don't know. We don't have. We are not Amazon. We are not Amazon. So if everybody try to access that, may take longer to load. But you can recall that URL and try later. And also the URL doesn't really work very well on mobile because we work with laptops. But once you get URL, you can move to a laptop and to play with it. And you can zoom in, zoom out, or just search your favorite gene to see the methylation of it. Yeah. Browser is nice, but we also want some metrics to, why is it not working? We can also look at using other way to look at reproducible, like correlation between different samples. So on the lab, I'm only showing 4 samples because we have so many data. So this is a subset of our all data. So the correlation basically means one sample on x-axis, one sample on y-axis. If it's perfectly correlated, it should be a straight line going from 0 to 1, right? But from the heat map, we can see its correlation is pretty well. And we are using two metrics to represent the correlation. One of them is called MAE, mean average error. So the lower, the better, right? The error is lower, the better. Another is the correlation. I mean, it's Pearson correlation. The Pearson correlation, the perfect Pearson correlation is 1. So straight line is 1, so the higher, the better. So we're using these two metrics to measure it. But this is only a subset. If we plot all of them together from MCF7, I don't expect you to read all the metrics because it's impossible, but just look at how perfect the heat map shows. Like all of them, like across different samples, across different sites, across different replicates. They all look very good, the correlation. And this is only MCF7. Sorry, why is it? And then we look at correlation between all of them. And I'm plotting the MAE number there, but you can't read the number, but you can look at the color. If it's red, means it's very, very low. And from here, we can see if it's the same sample, the correlation is very good. The error is really low. And between different samples, of course, they're different. Another thing I would mention is we found the non-methylated standard actually have some methylation. So this is still a subset of correlation from methylated on the right and the non-methylated sample on the left. And as you can imagine, if it's perfectly methylated, it should be everything about 100% methylated. So if you make the correlation heat map, just everything gonna group on the top. That's kind of look like that for methylated. But for non-methylated, it doesn't really group everything around 0. We can actually see a line in the middle. So that means for the non-methylated standard, actually it's partially methylated and it's reproducible between different methods. And actually, I think it's not really surprised. If you look at Zymo's FNQ, they actually said there are some residue methylation there, and they have a whole genome amplified different standard that should get rid of that. But this is the data we got here. And another thing we can look at data is we can make PCA plot. So from here, we have all sample together. From on the top left is methylated sample, all of them here. On the top right is non-methylated sample, and MCF7 and 12A78 are kind of together here, but with some separation. And then we can zoom in them. If we zoom in, we can see really the separation between MCF7 and the 12A78. And from here, you can also see some separation from hi-fi methylation from everything else. And also hi-fi methylation seems to be a little more variable with 12A78 sample. However, you can't really read it, but the PC2, the separation between hi-fi and others, actually very small, only expand like 3 or 4% of variance. So even if the visualization makes them seem very different, but it's not actually that different. And also we have EPIC Array. First of all, the correlation looks very good between different sites, And the PCA plot also looks very similar. I'm not going through the PCA, but it's like actually feels like turned an angle, but basically it tells us the same information. So at last, though, we actually have long-read and short-read methylation together. And we haven't really done a lot of thorough work, but just by looking at the browser, looking at some spot, we found some discrepancy between long-read, in this case, hi-fi methylation, and the short-read methylation. I think it's very interesting. Just keep in mind. From here, you can see there's one CpG site, right? For all 3 short-read methods call it unmethylated CpG sites. But for hi-fi, it doesn't make any call on this position. And on another position, the hi-fi is going to call it 100% methylated. And for the short-read data, it doesn't really have data there. So what's going on there? So we look at the one on the right first. if you look at the reference genome, if you zoom in, you can see it's a CCC there. It's not CPG sites. And then when you run the standard Bismarck pipeline, if it's not CPG site, you won't even attempt to look at methylation. So that's why for whole genome bisulfite, it won't give any data about this point. But if you look at all the reads on IGV, you can see there's a mutation from C to G in this sample. So now you have CPG site if you look at all the reads. And remember, PacBio co-methylation on raw reads without making any mapping. So when they see the read, oh, it's a CG, let's make a call. And then they make a call, and they're going to have a result here. What about the other side? It's very similar, but reversed. It's a CG side on the reference genome. However, if you look at all the reads, the C mutated to T. So the hyphen just see TG side and then just gonna ignore it because it's not CPG site. But when you using whole genome bisulfite or Bismarck, when you're using Bismarck to co-methylation here, it's gonna say it's 100% converted and then just gonna call it 100% unmethylated. So this is a problem with the standard pipeline, right? So once you have the conversion, you convert it from C to U, and then the U is going to read as T. And also, it's not just bisulfite. The enzyme method is the same. It's going to convert C to U, and then the U is going to read as T. And then after you get all the reads, you compare to the reference, and then now you know which site has been methylated or not. So it can really compute non-reference CG methylation, and after the conversion, CG becomes TG. So if you have TG to begin with, it will just assume it's unmethylated with standard Bismarck pipeline. So the conclusion in the end, all 5 platforms can very reliably give in CpG methylation in human cell line, and all of them work pretty good. And the Zymo standard, a non-methylated sample, we found it's actually partially methylated. I think Zymo knows, and some of you may know, just a note here. And then the third generation sequencing, I think in this way, it has some advantage over the short read method because all the methylation calling is done on the raw read data, so you can get very accurate result, even if there's mutations. And for the future direction, we actually want to integrate the array data with our sequencing data together. Right now, as you can see, all of them are separated, all the QC results. It's not really integrated on our browser. And also, we want to downsampling and to look at the limits. And then there are more science we can do, right? We can look at phasing, how methylation different in different allele, so very fancy stuff. And then we can also look at methylation detection in different regions, in hard-to-map regions, in repetitive regions. And last but not least, we can look at differentially methylated core between different platforms. Because we are showing we're showing you that looks very similar, but there's still some difference. We want to dig into the difference to see why they're different. And with that, thank you very much. And I think we can take some questions now.
Molly, Charlie, Xiaoyu, those were great talks. Thank you guys. And you know, Molly didn't even kind of like go into this. You guys have to think, as far as the DSRG, we came up with this study kind of last year with ideas, and we didn't even actually like start talking to vendors until the fall and to get through all these preps. Not even getting Xiaoyu data until what, February maybe? So yeah, it was a big deal. And so on that note, I want to also encourage you. I'm Jessica Podner, in case I didn't introduce myself at the beginning. So find myself, Molly, any of us, if you guys want to join DSRG or you're interested in GBIRG, definitely come find us. Also, showing on the slides in between talks, there's a QR code. If you have ideas for a study that you think that would be fun for the DSRG to look at, feel free to leave a comment, and we would love to get your feedback. And from there, I would like to open it up for any of our speakers, if anyone has any questions. Also, for our lightning talk, if you guys have a question for Brennan, please ask away. Yes.
Question for Molly. Aside from what we've seen in the library prep and the
good. We're on. Yeah, that is an input. That's a great question. Thank you, Sean. Obviously for the Illumina, the short read sequencing libraries, we were able to use, I think we used, oh my goodness, I'm blanking, 500 nanograms, 250?
Yeah.
Oh God, now I'm blanking. Now I feel silly. I think it was 500.
Yeah, it was 500.
500 nanograms. So that's, I mean, I feel like that's a doable amount. We did also try 10 nanograms for NEB. So we're kind of also working on that, kind of a little side project. But as some of you may know too, PacBio and ONT, they do require much larger input. So that is honestly where I think a lot of our kind of cost in like kind of dealing with all the large DNA, like we needed 5 micrograms per site for PacBio, and I think it was very similar for ONT. Yeah, I would like to see maybe doing that with a little bit lower input. So hello vendors, hi. So maybe we could do that, but I know that's also a treat on the long native sequencing. What's up, Claudia?
Actually, now that I think about it, was 100 nanograms.
100 nanograms.
Yeah.
Okay, okay. Thank you for the correction.
Sorry, I read the genomics course, like they start every day at 500 nanograms, it's like 4 kits.
Yeah, yeah. We wanted a standard, and we went with kind of what their input was. So, yeah.
And even some ERBSs, that's a different assay. Like we go down to 2.5 nanograms, and that some people are like, that's all I can do. Depending on who you work with. Can I ask a question about the computational side?
Yes, please.
So it's a good point about this part. Have you looked at other pipelines? I'm assuming for short reads they all depend on the reference genome.
We can also try. I think it's called BS-Seq. There's another pipeline. We can try, but I don't think that one can also look into the reference difference. Yeah, we're just getting started. So everything's on the plate, and we are exploring different options. Yeah.
Because that's a good observation that we never even think about. So I've slacked my advice.
If anyone else has a question, will you guys jump on the mic so that everyone can hear your question? Can you jump on the mic so everyone can hear you? Thanks.
Your mapping to your own genome browser is basically you still use HG39. I think currently more and more people trying to use T2T, which can give you more reference. I can notice there is a gap between. So in future study, compare short reads to long reads. I think T2T reference is a fair comparison for short reads because we can recover more information even from short reads.
Yeah, I agree. I think we can do that too. We can just swap reference using the CHM13.
Yeah, as T2T. So basically, we can detect 10% more CPG size if we switch to T2T reference. And also, second things about very interesting, the genetics. If they do have mutation, we have been observed it for many years. I don't know I'm not bioinformatician, but my bioinformatician sometimes give me some solution is you can look at the opposite strand. So basically, if you have a C mutation, sometimes, yes, it convert to T, but on the opposite strand, if it's C2T mutation, on the opposite strand would be A, so you can recover information. It's a little bit complexity, but certain clinical assays do require you do multiple way to separate so somehow stranded library is preferred.
Yeah. I think I got that too. If it's a stranded library, it's actually possible to distinguish that because the C2T conversion only happened one the other strands is not the
other
same.
strand can give you information. So do
Yeah.
you have like C2T is quite often mutation, so we can definitely try to aid like a pipeline. We work together too with DSRG as have more throughout digging more data from short reads.
Definitely. All the data present here is just we use the standard pipeline. Most people use like Bismarck and then run it. I'm sure there are a lot of
still very powerful, very powerful. I like it. Yeah. Thank you.
Yeah, thank you.
Yes.
Hi, I'm Chad Gieringer with Biomodal. We recently launched a 6-base solution, so you can do genetics and epigenetics in the same sample prep or short reads. My question is, how do we get involved and increase the vendor support amount for this project?
That's a great question. That QR code that Jessica was talking about, you can drop an idea in there. We love new studies, and if you're willing to support us, we're always happy.
Fantastic. Thank you.
Question for Brennan. I mean, you told us about those 3 different kits. Can you also elaborate on the cost and the time in the protocol, hands-on time on each of these kits, and any differences between them?
So the question was for the hands-on time in each protocol. I did find that Watchmaker was the shortest. That was all completed within 1 day. And then after that, I believe QuantaBio was up next, and then the Takara prep took the longest, but I also separated that out into 2 days.
And cost-wise, where everything?
Cost-wise, I would say that might be a question for Marie.
Yeah, so I will say, speaking of support, we got great support from all of the vendors that Brennan worked with for this. I should probably also use, I'm being a bad president here. So I will say we got great support from all of our vendors for Brennan's project. So we don't actually have exact costs for this. I will say one of the reasons that we were evaluating this study was simply because the process that we were currently using does tend to be expensive, and there do tend to be cheaper options available on the market now.
Thanks. Awesome. Any other questions for our speakers? Yes.
I guess this is to all of you, but from a vendor side, thinking about your bioinformatics solutions, not only for what you guys do in the core, but also in supporting your customers that are sending you samples. How are you guys thinking about data analysis and moving data around and sharing data? Are you guys looking into cloud solutions? Like, do you want things deployable only on local servers? Like, any insights you can give us on what would be useful for you?
We need data storage.
We need data storage. Yeah. Like, I think that's the biggest thing is where do you store it all? Like, I think we have transferring abilities. You know, we have that, but I think it's the storage. I mean,
this study alone, I think, what did it end up being? 30 terabytes.
Yeah, it's a.
Our Google Drive isn't 30 terabytes. So.
Yeah, I think the way we move data is actually fine. We are using globals to share data. And then for the data, everything presented here on the browser, because it's processed, it's a bad file like. So it's not too big, so I think I can put that on the STP server so you can see. But if you want to share all the band file visualization together, it's impossible to do that. We do want some like cloud solution. I worked with Envo before. I'm thinking about cloud-based computational platform. And also cloud-based data sharing platform. It's going to be very helpful for our future. Yeah.
In our operation, we were very focused on local HPC and have it migrated to the cloud. I've been doing all of our stuff locally. So.
Same, but it's also getting our clients to know how much data and where they're going to save their data. That's a struggle. They're like, I didn't know it was going to be this big. And I'm like, well, it's problem number 1. So, but that is, I mean, it's a good question. Do I have any answers, or no? I just know we need it. So.
Yes.
Sorry, can I ask the same question again, Charlie? In terms of cell numbers going into each of the different fixations and things, were they fairly similar, or did any of the approaches require more or less cells?
It's a little bit more of what?
Yeah, the cell numbers going into the preps, we tried to take the same cell numbers into all the preps. But this was a fixate, well, kind of, right? Because the honeycomb hive is kind of different than the flex protocol than from the parse protocol. But, the same number of cells were fixed for each site, and then each site got the same number of cells. Does that make sense? Yeah. Anything else? Awesome. Well, I think we're about out of time. Thank you guys so much for coming to see our talk.